Thank you. I'm Sandip Patel, and on behalf of SWOG, my co-investigators Young Chai and Rizal Kurzrock, I'm honored to present the results of the neuroendocrine cohort of S1609, DART, investigating ipilimumab plus nivolumab across a variety of rare tumors. These are my disclosures. And so 2018 has really been a watershed year, and as we continue here in 2019 for cancer immunotherapy. The Nobel Prize in Medicine last year was awarded for the discovery of the 2 immune checkpoints that we're investigating in this study. And ASCO for 2019 has named the Advance of the Year Progress in Treating Rare Cancers. In the spirit of these 2 intertwining concepts, we envision DART as a clinical trial to investigate the types of rare tumors that previously and historically have not been investigated for immune checkpoint blockade. Today I'll be focusing on the results of our neuroendocrine cohort. Overall, rare cancers are individually unique, but in aggregate represent almost a quarter of cancer burden. And so one of the issues with clinical trials in particular in this arena has been that these tumors are individually quite rare, though in aggregate represent a quarter of cancer burden, but also segregate across a myriad of histologies. This, in fact, is a common problem also with research in neuroendocrine tumors as well. And so SWOG S1609 DART is the first NCI-funded rare tumor immunotherapy basket study with over 37 rare tumor types being currently investigated. The primary objective of our clinical trial is to measure tumor shrinkage, overall response rate by RECIST 1.1 by rare cancer subtype. The regimen that we use for immune checkpoint blockade is slightly different than the ipilimumab-nivolumab regimen that's utilized in metastatic melanoma. We utilize a lower dose of ipilimumab, 1 milligram per kilogram given every 6 weeks, with nivolumab, 240 milligrams given every 2 weeks continuously in our study. Neuroendocrine neoplasms, which is the cohort I'll be discussing today amongst the basket study of DART, are a heterogeneous group in and of themselves of rare tumors that can occur throughout the body. While many are well differentiated and low grade, several, in fact, about approximately a third, depending on the organ of origin, are poorly differentiated or high grade. And these are most common in the lung, GI tract, gynecologic tract, as an unknown primary. These have an aggressive inherent biology due to their high mitotic rate, cell growth rate, and have few treatment options out of cytotoxic chemotherapy. Prior studies of anti-PD-1 monotherapy, our study utilizes anti-CTLA-4 and PD-1 combination immune checkpoint blockade, but prior studies of anti-PD-1 monotherapy in high-grade neuroendocrine carcinoma had an overall response rate of approximately 5%. These tumors are classified based on the primary site, for example, pancreatic, small bowel, gynecologic tract, and as well as proliferation index. And so in our study, we had 32 eligible patients, and I want to point out that pancreatic neuroendocrine cohort, due to its unique biology, is a separate cohort of DART that's currently accruing. And compared to the abstract that you may have seen, there's one patient that was re-stratified and found to have a pancreatic neuroendocrine cohort. Within this cohort, 56% of our patients had high-grade neuroendocrine carcinoma, of which almost half had a gastrointestinal primary, and approximately 1/5 had lung as their primary site disease. These are patients who are heavily pretreated with prior cytotoxic chemotherapy, with a median number of prior lines of therapy as 2. When we look at the waterfall plot for the overall response rate by primary site of origin, we did not see a particular trend for response in patients who received combination ipilimumab plus nivolumab. However, when we looked by tumor grade, regardless of primary site, it appeared that the majority of patients who could benefit in terms of tumor shrinkage in our study had high-grade neuroendocrine carcinoma, though there were patients with high-grade disease that did not receive benefit on the study. One of the salient characteristics of immune checkpoint blockade is the potential of durable responses. And this is something that's been seen in our study with patients with highly refractory disease on our study having responses lasting currently in the years. In fact, several of these are my patients in clinic, and it's particularly rewarding to see them continue to do well with an otherwise very aggressive disease that they've previously been treated with chemotherapy alone for. And so our overall response rate in high-grade neuroendocrine carcinoma was 44% independent of primary site, while patients with low and intermediate grade tumors had a response rate of 0%. We saw responses across the cohorts, whether it's a pulmonary primary, GI primary, or gynecologic primary, with improvement in survival manifested both in clinical benefit rate, 6-month progression-free survival, and median overall survival. Though these data are limited by the small numbers of patients we have and the fact that our clinical trial is not randomized. This was a well-tolerated regimen overall for our patients, with liver function abnormalities being our most common high-grade immune-mediated adverse event, colitis representing relatively rare toxicity at approximately 6%. We had no cases of pneumonitis and no fatal toxicities in this cohort. The predominant limitations of this study are there was no central pathology review. So adjudication of grade and organ of origin reflects real-world practice with primary sites having made these determinations. Also, our high-grade versus low intermediate grade analysis was not pre-specified, though we will have a high-grade neuroendocrine carcinoma cohort as a separate cohort pending to focus on this tumor type in which we've seen interesting biology. And then this is a single-arm cohort. It's non-randomized with 32 patients. And so the data are limited by smaller numbers in a non-randomized fashion. And to summarize, DART S1609 is the first NCI-funded rare tumor immunotherapy basket study, which we believe is unique in its design and scale. And we're studying over 37 rare tumor types with a combination of ipilimumab plus nivolumab. The neuroendocrine cohort here, the non-pancreatic neuroendocrine cohort, had promising signs of benefit in patients with high-grade neuroendocrine carcinoma independent of primary site, with a 44% overall response rate in high-grade disease compared to no responders in our cohort with lower intermediate grade disease. Additional analyses are underway. We will have a confirmatory high-grade only neuroendocrine carcinoma cohort in DART upcoming, and as well as the translational analyses from the specimens for these patients is underway through the NCI CMAX mechanism with Dr. Wastuba. And more broadly, one thing we wanted to really reiterate here as the investigators on this study is that clinical trials in rare tumors are feasible. There is a mantra that rare tumors clinical trials are not able to be conducted successfully, and it's led to some historical inhibitions in pursuing these studies. This particular study is open at 800 sites throughout the country, and almost 2/3 of the patients in this cohort were treated at community sites close to their home. We've currently had 560 patients enrolled in our rare tumor study across our 37+ cohorts within the past 2 years, and this particular cohort accrued within 3 months. And so we hope to show that rare tumor clinical trials are not only feasible, but have potential for benefit for patients with rare tumors who represent in aggregate almost a quarter of patients we treat with cancer. And with that, I'd like to thank the wide team all throughout the country that's taking care and helped put this protocol together and taking care of these patients, including my co-investigators, Dr. Young Chai and Rizal Kurzrock, the teams at SWOG and CTEP, as well as our CMAX collaborators, and most of all, the patients and caregivers for trusting us in the conduct of this study. Thank you.